Hello, bacteriophage scientists. Welcome to another week of pre-open lab videos. This week, I'm going to go over your Streak 2 protocol. It's not going to be a lot of new things. Actually, a lot of it is just going to be very niche things that you need reminders on for this week. But the specifics of the protocol, how to do the streaking, how much bacteria to add, all those like very specific things are going to be in last week's video because you'll notice all of this is just a repeat. So if you need a refresher on the protocol, in more specific detail, I implore you to look back to the Streak 1 video from last week, or also looking at the lab manual pages 33 to 34, and then 105 through 109, because they're the exact same, okay? So let me talk about the lab notebook for a little bit, because it's a little bit different this week as compared to more traditional weeks, in that we're doing this over a course of 3 weeks. So the streaking protocol in your lab notebook is not necessarily going to be finished until you complete all 3 weeks of it. So you can think of a protocol as not being finished until the objective is reached. So what is our objective with streaking? It is to obtain a clonal population of phage that we're isolating from a single bacteriophage plaque. So that's not going to be done until we're 3 weeks in, okay? So you're not closing out this protocol necessarily in your lab notebook for 3 weeks, right? You're not going to have your all of your results. You're not going to have your analysis and interpretation, and you're not going to have your next steps necessarily until those 3 weeks are done. So you should still be taking photos of each week's streak plates and uploading them into your lab notebook, because that's very important. And when it comes to tracing the history of that phage sample, right? So looking from your first plaque, from your direct or maybe your enriched isolation, all the way to the third streak, what differences do we see? Okay, so I have an example trace history path here below, where all the way in the left, this is a mock direct isolation plate, right? Looking at it, you can see it looks like there's different morphology. Some are clearish, some are cloudyish. They could be different species, we don't know. So if we streak it across, for this example, 2 weeks, and our resultant plate looks a lot different from our initial plate, right? We're looking like all of these plaques on the right are all the same size. They look like they're the same color, same morphology, right? That's probably telling us that based on this plate on the right and looking back relative to our first plaque in our direct isolation plate, we produced a clonal population, right? These look like they're all the same. We likely have one species of phage that we're working with, which is very important moving forward, okay? It's also very, very important that at any point throughout the semester, but specifically we're talking about this 3-phase streak week, that if you are switching samples, if you're borrowing samples for streaking in any way, that you are obtaining location information for those samples if you're not streaking your own samples. Okay, this is going to be very important when it comes later on into your paper, but relatively speaking, if someone is looking at your lab notebook and wants to reproduce your work, they need to have a clear understanding of your sample and its history and where it's coming from, okay? So asking whoever you're borrowing from for some information is very, very important. Let's go over streaking. Again, this is not going to be a very detailed elaboration on what to do, because you've already done this week. If you need a refresher on that, watch the Streak 1 video, but this is going to be the more nuanced details of this week specifically. So one, what type of plaque are you going to be picking to continue streaking? Okay, so you're going to be looking at your plates. An ideal plaque, same as last week, is going to be a well-isolated one. Ideally, if the dilution worked on everyone's plates, your section 3, your third streak section, would be the ideal place to look for a nice isolated plaque. But you'll find that that might not necessarily be the case. They might be in your streak 2 section, maybe even in your streak 1 section. You might have to just find a nice isolated plaque to streak, okay? So it'll be the same deal as last week's, make sure you're circling and taking the photo, putting your initials, all that good stuff. So if you are using if you only have one successful plaque or one successful plate with a plaque, you're just gonna take one plaque and you're gonna streak 2 separate plates. Same plaque, 2 plates, okay? Same as last week for the majority of you. If for some reason you are working with 2 different samples, 2 different soil samples on 2 different streak plates, maybe your direct isolation plates produced really good results and you just really want to streak those through and you're working with 2 samples, you can just continue doing that. Streak 1 plate from those individual unique samples if that is the case, but I believe a majority of you are just streaking one singular sample. If you didn't get any streak results for any reason, maybe you were too aggressive with the auger or forgot to add bacteria, whatever didn't work for some reason, or you were quarantining last week or couldn't come in because of any like health related reasons or anything like that, and you don't have a streak plate to streak your streak two on, we are just going to borrow a plate, likely from someone in the class that has streaked a plate and was successful in producing plaques. You're going to borrow a plaque from someone and it'll be the same deal. You're going to circle one, you're going to put your initials, you're going to take a photo, and you're going to streak 2 plates from that singular sample. Okay. And then make sure you are getting that location information. I really cannot emphasize this enough. You're getting location information and you are going to ask for photos. So when I say ask for photos, what I mean is that you are going to be given a streak one sample. You're going to take a photo of it and you're going to streak your streak two, but it's important that you know where that initial streak plate came from. Did it come from a direct isolation plate or an enriched isolation plate? Okay, get a photo of that plate. That way you can trace the history, morphologically speaking, of that particular sample. Okay, in the backup plates we give you, there is a lab notebook in 330 and the lab bench in the front of the classroom that has a bunch of backup plate information on it. Also, there is a link on Carmen under lab resources that takes you to a community plate Excel sheet that has all the info for the backup plates that we've been using in this class, along with, if it's another student, a point of contact. So you can contact them via email and ask them for photos or any information that they may have if it's not on the sheet. Okay, very important. Do this in the classroom. Okay, don't wait to do it outside of the lab while you're with that person. Just exchange photos, email, iCloud, drop it, whatever. But very, very critical, especially later. So let's briefly talk through the most important aspects when we're talking about streaking. Make sure we're picking an isolated plaque if possible. That should be a main priority for us. Make sure we're circling it, we're writing our initials around that sample, we are taking a photo for our lab notebooks. Get a fresh stick with every single streak. This was a little bit of a problem last week where people were using the same stick without realizing it. Make sure we're paying attention, getting a new stick each time. Pour the top agar and bacteria solution onto your plate immediately in that little third phase streak area where you draw your X or put a little dot, however you denote it, immediately. This should not be something that you are waiting or delaying to pour. There's no inoculation step. With a bacteria and agar, you are pouring it immediately. Okay, if you delay, this is why we see issues with the agar not solidifying, with tears in the agar, with it not covering the whole plate because it's solidifying in your tube as you're waiting for something. Okay, another pro tip here is when you're getting that top agar out of the water bath, give it a little swirl. Sometimes the liquid media in that top agar and the agar particles can separate if they've been sitting out too long. So you might just be sucking up liquid and not actually the agar. So make sure just a little gentle swirl, nothing too aggressive to mix that up. Okay, pour your top agar onto the actual agar, not the lid. We're paying attention to that. And let's try to be a little bit less aggressive with the agar. This is everyone's first time, for the most part, last week, streaking on an agar plate. Let's try to be a little less aggressive, a little more gentle with our streaking. Okay, we don't have to be, too extreme. I promise you're getting those plaques on there. So let's relax our hands a little bit. Now let's talk about some bad lab habits that we really want to eradicate this week. Hopefully, as we're coming into this week, we are familiar enough with aseptic technique and the expectations of aseptic technique, but let's talk about some bad lab habits that we're seeing collectively across the class we want to just eradicate completely because they are leading to contamination. So one, placing the Petri dish lid onto the lab bench. This is probably the biggest issue we're seeing. You should not be placing any lids on your lab benches because they are contaminated areas. Even if you are lysoling them, you're introducing bacteria with your person, any materials you're bringing into that lab bench. So do not place it on the lid, right? Luna the lab corgi is not pleased with this. Bacteria is everywhere, you're gonna contaminate. You should be holding the lid at all times and within your aseptic zone. We see people holding the lid, but also moving 6 feet away from their flame. So that's contaminating it still. You shouldn't be exposing that auger to the air for an exceedingly long time. So always, if you're working near your flame, holding that lid like so. Another one is placing top auger bottle lids on the lab bench. We don't want to see that. Luna lab corgi, not pleased with that either. Okay, because again, bacteria on your lab bench, and this is a solution that you are contaminating for everyone. Okay, if you accidentally contaminate the top auger and then put it back in the water bath, contaminate it for everyone. Okay, so don't be that person. Practice good aseptic technique. Hold the lid, work near that aseptic zone at all times. Similarly with the bacteria tubes, the lid should not be going on the lab bench, okay? You are contaminating it and you're contaminating it for everyone if you do this. So similar to the top auger bottle, near the flame within that aseptic zone, holding the bottle lids, or I'm sorry, the tube lids, like so. You're not moving 10 feet away from the flame. Everything is gonna be within that zone. Again, make sure we're passing glass tubes through the flame or flaming them very quickly. You don't have to hold it. You shouldn't hold it in the flame because you're killing anything in that tube if that is the case. But just a quick, just like bloop, bloop, through the flame before and after you add something to it. Very quick. And again, I really sound like a broken record here, but we're seeing this again and again. Do not let your top auger and your bacteria solutions sit in those tubes for any period of time. There is no inoculation step. As soon as you add your top auger, you are pouring it directly into that dish. Okay? If you let it sit for too long, things like this can happen. So hopefully everyone will take that to heart and practice safer and better aseptic technique this week to reduce contamination, because we did see quite a bit of contamination across people's plates last week. So let's try to eradicate that as best as we can. This week lab again will be 1 hour. So you should be signing up for a 1 hour open lab session. Ideally do it as early as possible to make sure we're getting the seats filled to your particular schedule desire. But if you have any questions, you can reach out to your TA, you can reach out to me. Happy to clarify anything. And I wish everyone the best of luck in Open Lab this week.